Hello everyone, welcome to the new video tutorial of Bio. In the last session we have dealt about the second portion of cell biology, and that is biochemistry of cell. We have discussed only a brief portion of it. Now in this session we will be continuing the third part of the cell biology, and that will be discussion of the colloidal nature of cytoplasm. If you haven't subscribed to our channel, subscribe to it and enable the bell icon to get daily notification. So without wasting time, let's get on with the video. Before discussing the colloidal nature of cytoplasm, first we need to understand what is a colloid, so that you get a base and foundation to develop the concept of colloidal nature of cytoplasm. You all know two types of mixtures actually, that is true solution and one is the suspension. True solution means the solute completely dissolves in the solution, and in the case of suspension, the solute does not dissolve in the solvent, and it settles down to the action of gravity. There is a mixture in between these two, and that is known as colloid. That the particles or solute in this one is too large to form a solution, but it is too small to settle under gravity. So this comes in between a true solution and suspension. So actually the particles, colloidal particles, are actually larger than the solute seen in true solution, and a little bit smaller than the solute that we have seen in the suspension. So that's the first point. And the particle size which we normally see in a colloid ranges between 1 by 10 lakh to 1 by 10,000 millimeters. So that's also an important point. An example we to see to understand what is a colloid is a best example is milk, cheese and egg white. These are some of the examples that we have to we can see in a daily life as colloids. So colloidal particles are in between, the size of the particles in between the solution particles seen in true solution and that of seen in a suspension. So that is a base or understand the colloids in a simple way. Now let's discuss the nature of a colloid. To understand what is a colloid, we need to understand the nature of colloid also. So there are two phases seen in a colloid. The first one is dispersed phase or discontinuous phase. So it's also known as discontinuous phase. And second one is dispersion medium. The dispersed phase, it is actually the particles which we see of colloidal size. The particles of colloidal size are known as dispersed phase, and it is actually dispersed in the medium known as dispersion medium. So taking the biological example, it can usually dispersed phase consists of macromolecules like proteins, fatty acid, nucleic acid, etc. And the particles are known as micelles. So that is an important point. The particles in dispersed phase are actually known as micelles. Now dispersion medium, it is the medium in which the particles of colloid size are dispersed. So that is the point, that particles of colloidal nature disperse in a medium, and that medium is known as dispersion medium. Since these particles of dispersed phase are known as micelles, the dispersion medium particles are known as intermicellar particles. So that is also an important point concerning to the nature of colloid. So there are two types of phases, dispersed phase and dispersion medium. Dispersed phase is actually the particles of colloidal size, whereas dispersion medium, it is a medium in which particles of colloidal size are dispersed. So that's the 2 points that we have to notice here. Now let's discuss the colloidal nature of cytoplasm. Coming on to the colloidal nature of cytoplasm, it was proposed by Fisher and Hardy. So in concerning the entrance is a really important point, that Fisher and Hardy proposed the colloidal nature of cytoplasm. Now in cytoplasm, the colloidal particles are actually large size proteins along with tiny droplets of fat which is scattered in the medium, that is a water. So that is a point should be noted, that the colloidal particle dispersed phase is the large proteins and fat droplets, and the dispersion medium is the water. So that is the colloidal nature of cytoplasm. Now the colloidal nature of cytoplasm gives us some properties, and the properties are described here. The first one is that it gives electrical properties of cytoplasm. There is a Brownian movement, that is a zigzag movement of particles in that. And also the Tyndall effect. Tyndall phenomenon is actually the scattering of light by the colloid. So that's also observed in cytoplasm. And finally, sol-gel transformation. Actually, in all organisms, the cytoplasm is a gel form. It is mostly seen in gel form, but when the temperature increases, the cytoplasm actually changes salt form, solution form. And when temperature normally decreases, it transforms back to gel. So this sol-gel transformation is seen in cytoplasm. So these all properties contribute to the colloidal nature of cytoplasm. That's the important point. So this should be noted here, that is the gel is normally in organisms, in all living organisms, the cytoplasm is in the gel form, but when temperature increases, it transforms into solution form, and when temperature decreases, it returns back to gel form. So that is an important point to be noted here. So that's all we have to discuss regarding colloidal nature, cytoplasm and biochemistry of cell. If you have any doubt or confusions, please mail it bio@gmail.com. Wait till next video.